Welcome. In this video, we're going to explore transcription, the process of transcribing or copying the genetic information stored in DNA into messenger RNA. The template strand of the DNA is read, and mRNA copy is built from it. It transports the information encoded in a gene out to the ribosomes. The region of DNA being transcribed, called the transcription unit, includes a promoter, an initiator, a terminator, and introns. The very first mRNA produced before any editing is called the primary transcript. There are 3 DNA-dependent RNA polymerases. Polymerase I, or type A, makes ribosomal RNA. RNA polymerase II, or type B, is the main enzyme. It's the one that makes mRNA. And RNA polymerase III, or type C, makes tRNA. Before transcription can begin, RNA polymerase needs to find the right spot. Consensus sequences on the DNA act as start signals, sitting upstream or downstream of the actual start site, and RNA polymerase attaches at a promoter site on template strand. Transcription rate is also tuned by regulatory sequences. Enhancers increase the rate of transcription while silencers decrease it. Hormone response elements are another kind of regulatory signal, and insulators sit between silencers and enhancers, preventing interference with each other. At least 7 transcription factors, collectively called TF2, help assemble the machinery. TBP, the TATA binding protein, recognizes the Goldberg-Hognes box, while SL1 helps RNA polymerase locate the exact start point. TBP-associated factors then join TBP to form TFIID, and its binding to the TATA box is the first real step in transcription. Other proteins, TFIIA, B, E, F, and H, together with polymerase II, then join to form the pre-initiation complex, built from 3 classes of factors: basal components, co-regulators, and activators. Once initiation is complete, elongation begins. RNA polymerase moves along the DNA template, incorporating new nucleotides into growing mRNA one at a time, following the base pairing rule. The new mRNA strand is synthesized from its 5' to 3' end, which means the template DNA is being read in the opposite direction. As RNA polymerase moves forward, it continually unwinds the double helix with help of topoisomerase, which relieves the strain of unwinding. This creates a transcription bubble, roughly 20 base pairs long, containing the polymerase, DNA, and nascent RNA. Because RNA polymerase has no proofreading ability, its error rate is about 10,000 to 100,000 times higher than DNA replication. That's less serious than it sounds though, since these mistakes are never passed on to daughter cells. Transcription ends when a termination protein, the Rho factor, recognizes a specific signal. A GC-rich palindrome sequence forms a stem and loop structure, immediately followed by a run of 6 or 7 U residues in the RNA chain. Attachment of the Rho factor is an ATP-dependent process. Once it attaches, RNA polymerase can no longer move forward, so the enzyme dissociates from the DNA and the newly made mRNA is released. mRNA that's formed and released from the DNA template is called the primary transcript, or heteronuclear RNA. Before it can leave the nucleus, it undergoes extensive processing in the nucleoplasm to become mature mRNA. The first modification we'll look at is poly-A tailing. The 3' end of the transcript is polyadenylated right in the nucleoplasm. This poly-A tail can be anywhere from 20 to 250 nucleotides long, and its job is to protect the mRNA from being degraded by 3' X nucleuses. Every eukaryotic mRNA is also capped at its 5' terminus by a molecule called 7-methyl-guanosine triphosphate. This creates an unusual 5' to 5' triphosphate bridge. This cap is what the translating machinery uses to recognize the mRNA as ready for translation. Methylation is another common modification. The N6 position of adenine residues and the 2' hydroxyl group of ribose are frequently methylated out in the cytoplasm. primary transcripts are very long, with molecular weights over 10 million, while the mature mRNA is much smaller, around 1 to 2 million. That's because large portions of the hnRNA are cleaved away. The primary transcript contains coding regions called exons interspersed with non-coding regions called introns. These introns are cut out and exons are spliced together to form the mature mRNA. This all happens in the nucleus, and splicing is an energy-requiring process. Because transcription is so essential, several drugs work by blocking it. Actinomycin D and mitomycin both physically blocks transcription and are used as anti-cancer drugs. Rifampicin, meanwhile, is widely used in treating tuberculosis and leprosy. So, to bring it all together, transcription moves through initiation, elongation, and termination, followed by post-transcriptional processing to produce a mature mRNA